my name is Claude Perreault. I'm a professor at Université de Montréal and PI at the Institute for Research in Immunology and Cancer. Over the last decade, a main endeavor of our team has been to elucidate the biogenesis of MHC-associated peptides, or MAPs. MAPs collectively form the immunopeptidome and regulate all events in T-cell development and function. From a medical standpoint, MAPs present on cancer cells are of prime interest because they represent potential targets for cancer immunotherapy. Our approach for the discovery of actionable tumor antigen has two main features. First, its scope. We search for tumor antigen coded by any reading frame of the entire genome. And secondly, our method. We don't merely predict the nature of tumor antigens. We identify them by direct mass spectrometry analysis of primary tumors. This video will summarize our recent work on the immunopeptidome of breast cancer. This study yielded unexpected insights into the immunology of breast cancer and was expertly spearheaded by my colleague Eralda Kina.
Hi, my name is Eralda Kina. I'm an oncologist and a PhD student here at the IRIC. Breast cancer is the most prevalent cancer in the world, and breast cancer subtypes are quite different from an immunological standpoint. Indeed, hormone receptor positive breast cancer is lowly infiltrated by T cells and are considered cold tumors, whereas triple negative breast cancer tumors are highly infiltrated by T cells and are considered hot tumors. So what exactly happens to explain this phenotype? And is it possible that the antigens presented by those two subtypes are different? That is what we aim to investigate in this project. In cancer, we are interested in two types of antigens. The first ones are tumor-specific antigens that we can only find on tumor cells. They can either come from mutated regions or aberrantly expressed region. The second type of antigens are tumor-associated antigens, which can be found on normal cells but are overexpressed on cancer cells. So we applied our proteogenomic approach using mass spectrometry, RNA sequencing, and bioinformatics in order to identify and classify our antigens of interest. We identified more tumor-specific antigens in triple-negative tumors, as you can see here in blue, which provides a rationale for their increased immunogenicity in vivo. We found an equal number of tumor-associated antigens in both subtypes, and most of our antigens were novel. Most of our TSAs derived from cancer testis antigens or non-coding regions. Using the TCGA cohort, we saw that triple-negative tumors express a higher level of tumor-specific antigens than hormone receptor-positive tumors. We also saw that predicted presentation correlated with immune infiltration. We identified quite a few TAAs in both subtypes. Of interest were a group of TAAs that seemed to derive from cancer-associated fibroblasts. In order to confirm this, we used single-cell datasets that were already published, and we evaluated if the source genes indeed seemed to derive from cancer-associated fibroblasts. And we found that yes, this was the case. This is an interesting finding, as this can be a new type of vaccine target. Using FAST assays, we saw that many of our antigens were immunogenic in vitro. We wondered if the predicted presentation of our antigens impacted overall survival. Again, using the large TCGA cohort, we calculated the number of antigens presented by each tumor. And we found that for triple-negative tumors that had a high level of predicted presented tumor-specific antigens, they had an overall survival benefit. This was the case for TSAs that derive from non-coding regions and cancer testis antigens. Of note, this benefit is HLA restricted, suggesting that it relies on CD8 T cell activation.
Immune checkpoint blockade certainly represent a seminal breakthrough in cancer immunotherapy. However, considering all the checks and balances in the adaptive immune system, I doubt that immune checkpoint blockade will suffice to cure most cancer types. I rather think that progress in cancer immunotherapy will depend on antigen-specific approaches. Since antigens reported in this study were shown to be immunogenic in vitro, they represent potentially attractive targets for antigen-targeted cancer immunotherapy based on vaccines or TCR-based biologics.
We would like to thank our collaborators here at the IRIC for their help on this project.
And we are grateful to the Canadian Cancer Society for funding this work.